I would like to also introduce the first presenter of the day, Alireza Akhavan, the Provincial Specialist of Plant Disease with the Saskatchewan Ministry of Agriculture. Prior to joining the ministry in 2020, he was at the University of Alberta and Agriculture and Agri-Food Canada in Lacombe for almost 10 years, teaching plant pathology and working on various diseases of canola and cereal crops across the prairies. Alireza will be talking about identifying canola diseases today.
Good morning, everyone. Thank you very much, Tracy, for the introduction. So my name is Alireza Akhavan, and I am the Provincial Plant Disease Specialist for the Government of Saskatchewan since 2020. And as Tracy already indicated, before that I was with University of Alberta and Agriculture Agri-Food Canada in Lacombe for almost 10 years, working on canola diseases, mostly on clubroot and blackleg and sclerotinia, and also worked a lot on cereal leaf diseases, particularly barley net blotch and wheat tan spot. So today I am going to give you canola disease identification kind of instructions and also give you updates of what we have seen last growing season in Saskatchewan across all the regions. And I try to be, of course, canola diseases are very diverse, but I will focus on what is important on the prairies, and also I need to be like a little bit quick. I have 54 slides, but most of them are just pictures, so I am quite confident and I meet the time limit. So Saskatchewan had three main canola-related surveys last growing season. We have one general canola disease survey, which looks at all the diseases. In that survey, we'll look at prevalence, which is number of crops with disease, incidence, which is the number of plants with disease in each crop, and severity, which is the portion of the plant which is diseased. Because I am talking during my talk about prevalence, incidence, and severity very often. We also had one verticillium-specific survey after harvest on the extreme east side of the province. And we also had a clubroot monitoring program, and we looked at 521 fields across the province. So again, because this is canola diseases ID and update, I will go to the ID part for each disease first and then give you an update of what happened last growing season. So starting with sclerotinia stem rot, again, this is the name of disease, sclerotinia stem rot. The name of the pathogen is Sclerotinia sclerotiorum, and in this disease we have hard bodies of the fungus, which are like moss dropping, and they are black, and they are called sclerotia. So Sclerotinia stem rot is the name of the disease. Sclerotinia sclerotiorum is the name of the pathogen. Sclerotia is the name of the just structure of the fungus that you will see quickly. So in this picture, these are sclerotia of the Sclerotinia sclerotiorum, and these are apothecia, and we'll see them quickly. So this disease only has one cycle, so it has no non-asexual spores, so there would be no further spread of the disease within growing season. So there would be only one cycle. It starts from a stubble, and then plant get attacked, and that's it. And then we'll get the disease. So it is not something that happens multiple times during the growing season. We call them monocycle disease. And development of epidemics is heavily dependent on what you will have in the upcoming growing season. So it is very hard disease to forecast because they are heavily dependent on moisture. Like in 2021, which was a very dry year, almost we couldn't find any unless it was an irrigated field. And last year, especially on the east side of the province, which was wet, we had very many. So it's heavily dependent on what the environment brings us in the upcoming growing season. So it's a generalized disease cycle, so we simplify that. We start from here. These are the sclerotia within the stem cell within the stem on stubbles. So these germinate and produce these mushroom-type structures. They are a little bit smaller than a dime size. It should be like a size of your small fingernail. So if it's something big, it is not sclerotinia. If you see a mushroom, it's not sclerotinia. It should be a dime size. And you should see them if the field is infected around June, not later. And again, this structure, which is called apothecia, is the sexual structure, has lots of sexual spores called ascospores, and they can travel with wind. They have to fall on petal. So this is a different from sclerotinia stem rot on canola versus what we get in pulses. In pulses, the sclerotia can germinate and attack the root directly, but in canola, ascospores need to be fallen on a flower. If they fall directly on a leaf or a stem, nothing happens. They are not powerful enough to infect leaf or a stem. They need to infect the flowers, and then fallen flowers need to stick to leaf or stem to cause the disease, because on that time, flower provides enough energy to the fungus. Fungus grows, and then it's heavy enough to attack the stems. And this is one cycle, so that's it. And then you get the bleached stems. And if they are severe, at the end of the season, if you open them, you will see this moss or rat dropping kind structures called sclerotia. So this is the time that people should be concerned when you see flowers on the stems. So if the flowers are already infected, the infection can go to the leaf or to the stem. Usually if the flowers fall in the corner that the leaf attaches the stem, that's the best for the fungus because it directly gets to the stem. It doesn't need to infect leaf first and then gradually get to the stem. It gets to the stem, and then we get more severe diseases. This is a picture of end of the growing season when everything is just covered and colonized by this white fluffy fungus. So some reviewing of signs and symptoms of disease. Disease can be everywhere in the plant, so it can be found on pods, and this is the bleached area. This is a small stem infection. So if it is a sclerotinia stem rot, you shouldn't see any structure on the lesion. So if you see dots on the lesion, it is not sclerotinia. It is usually bleached with just darker margins. And then the fungus can get to the stem, becomes quite heavily. At the end of the season, the stems start to be shredded and shattered. Or if you just use your finger and open the stem, you will see these moss droppings, which are the sclerotia of the fungus, which can stay in the soil for up to 5 years, but 3 years for sure, they just stay viable, and they are ready to germinate whenever moisture is provided. So more photos of infection on pods, and you see one of those sclerotia on here, but this can be here, and a heavily infested stem. So again, there is no other structures. It is just a fluffy fungus if it is really moist. Otherwise, it is just bleaching a stem with darker margins. And again, this is a very severe case that the whole plant, almost from the base, is infected. And I'm quite sure if we open this stem, we'll see very many of those sclerotia. So when we are in the field, we also check for severity. So severity for sclerotinia is 1 to 5. So 0 is, of course, no disease. 1 is only you see disease on pod. 2 is if a quarter of the plant is impacted. 3 is half of the plant impacted. 4 is 3/4 of the plant is impacted. And if the disease is really on the base and impacts the whole plant, then it is just number 5. So last growing season on sclerotinia, more than half of the fields in Saskatchewan had the infection. So the prevalence was 56%. So 56% of crops surveyed had Sclerotinia in the province. Incidence was 6% when we averaged across the province. So again, incidence is the number of plants per field surveyed. And usually we look at 100 plants per field. So this is the average of 205 fields of incidence in 205 fields. So it's a very reliable number statistically. 11% is the incidence in infected field only. So 6% overall, 11% in infected fields. However, severity was quite low, 0.2 on that 1 to 5 scale across all surveyed fields, and 0.3 across infected fields only. Regional breakdown, as we see here, Northeast had the most crops with the disease, and East Central had the highest incidence. And as we see, overall regional breakdown is very well aligned with the weather pattern. So we had way less disease in Southwest, which was dry, and West Central. So the higher the moisture, more Sclerotinia. Very dependent disease on moisture, and very hard to predict what we get next growing season. So over years, again, these ups and downs over the years back to 1999, it just shows how dependent this disease on moisture. Like in 2016, which was a very wet year, we got up to 24% incidence. And like 2021, which was very dry, we got as low as 2%. And then again, this year we got back to 6%, so almost 3 times more than 2021, because it was moist in very many areas of the province. But of course, it was not as high as 2016, not as low as 2001 to 3, which was a very dry period of the time for the province. So that was about Sclerotinia. We move now to another very important disease, blackleg. Sometimes it impacts the trades, so we always keep keen eyes on blackleg, even though that breeders did a very excellent work on blackleg resistance. So we are quite on the safe side of this scale when it comes to blackleg. So blackleg is caused by 2 pathogens. Leptosphaeria globosa is a weak pathogen. We are not worried about that. They just can cause superficial lesions on the stems. The big guy is Leptosphaeria maculans. This is the virulent pathogen. This is the pathogen causes like base stem canker, and you see blackening of the cross section. This is Leptosphaeria maculans. And it can impact both seed quality and yield. So this is, again, generalized, simplified disease cycle. So I can perhaps start from the overwintering stage or post-harvest. When we look at the stubbles, there are some, again, sexual fruiting bodies on the stubble, and we'll see them shortly how they look like. And they're called pseudothecia. They produce ascospores, which are the sexual spores again, which they can attack seedlings. So usually up to 6 leaves, seedlings are very susceptible, and they cause lesion on seedlings. But this is a polycyclic disease. So on these lesions, you will see lots of dots. Each dot is a pycnidia, which is the asexual fruiting body, produces lots of new spores through asexual reproduction. They are called pycnidiospores, pycnidiospores, that they can splash through growing seasons. So under irrigation or rain and a storm, we'll get more and more disease. So that's the difference with Sclerotinia, which was just 1 cycle. This is a polycyclic disease. And then when the plants, the leaves, and stems get infected, then it finds its own way to the bottom of the plant and causes canker on the base. So we'll see more pictures. A variation happens from region to region, from year to year, but this disease is not easily impacted by environment. Again, opposite to Sclerotinia. So even on a dry year, we'll get lots of blackleg. So actually, it doesn't matter much for the disease, the moisture. And we'll get to some more information quickly. So looking at the signs and symptoms of the disease, so after harvest or before seeding, if you walk throughout the field and you see something like this on canola stubbles, lots of dots, which can be seen by naked eyes, but it's very easy to see them with just a normal magnifier. If you see something like this, you're most likely looking at black leg, so you should be more careful. So these are, again, pseudothecia, or the overwintering structure of the fungus that produces those ascospores, and those ascospores attack cotyledons and young leaves. This is how blackleg looks like on leaves and cotyledons. So usually from dirty white to brownish gray lesions, lots of dots on them. So there is no other disease like that. So if you see this is blackleg for sure. So you will see the spot or the lesion with full of pycnidia, which are the asexual bodies that just repeat the disease cycle during the growing season over and over. And again, no other diseases like that. If you see this, it is blackleg. So more pictures, and usually, again, these are from up to 6-leaf stage. Again, this is blackleg. Lesions, lots of pycnidia. That's only blackleg. So if you want to just know the difference between lesions caused by sclerotinia, because usually when we say sclerotinia, everyone think of stem, but sclerotinia most of the time start from the leaf. So if we want to compare the lesions of blackleg versus the lesions of sclerotinia, then on blackleg you see these small dots, and these are pycnidia. There is no other disease like that. For sclerotinia, however, first of all, again, I already said that you need these petals fallen on leaf. The ascospores or the fungus can infect petals first, and petals need to stick to a lesion, and the lesion should be water-soaked to kind of make sure this is a sclerotinia. So usually we have petal sticking to the leaf and water-soaked lesion, no structures, no dots on the leaf. So this is a sclerotinia, this is blackleg, quite very distinct lesions. So on the stem, of course, we have lesions, and this is the reason we say canker. Canker means lesion or wound. So if you see lesions full of dots, which are pycnidia, then that's blackleg. So these are, again, severe base stem canker, full of small dots, which can be seen easily with a magnifier. This is blackleg. And of course, if you have the cross section, you also see the black wedges there. We'll see better pictures. So when it comes to a stem, if you want to differentiate sclerotinia and blackleg, straightforward. Sclerotinia stem rot, no dots on the stem. Stems gets shattered and shredded. If you open it, you see big, like rat dropping size or moss dropping size structure called sclerotia. For blackleg, there is no shredding or shattering. There is lesion full of dots. And usually, if you look at cross-section, you also see that black wedge. So blackleg versus sclerotinia stem rot, quite distinct symptoms. Again, even if it is mild blackleg, still you will see these wedges of black discoloration. And of course, you never look at one symptom. You look at everything. So if you see the lesions there at the soil line, and then you cut it with your clipper, look at it, and you see this, then this is blackleg. But sometimes it's easy work because it's so bad. So it's so solid, and it's just blackleg. So if you get your clipper and you see this, that's blackleg. It's so solid, it can't be anything else. But blackleg can also be odd. So it can produce some even star-like structures or kind of lines and stuff. And these are all blackleg. But also try to look at if you see this lesion with pycnidia to just make sure that's 100% blackleg. Blackleg can be seen together with root maggot tunnel. It is not uncommon. So this is blackleg. Again, when we are in the field and we want to assess the severity, again, it's a 0 to 5, like a sclerotinia severity scale, with 0 absolutely healthy. If a quarter of the cross-section is blackened, then it is number 1, up to 50, number 2, up to 3/4, number 3. If the whole cross-section is discolored, but plant is in good shape, no constriction, no death, we give it 4. But if the plant is dead, we'll give it 5. So last growing season, blackleg was found in 73% of Saskatchewan canola fields. Incidence was 11% across all surveyed fields, 15% when averaged across infected fields. Severity was 0.2 or 0.16 across all surveyed fields, 0.22 across infected fields. So very high prevalence, very high incidence, but quite low severity. It shows the great work breeders did, actually. So very high number of crops have the disease, very high number of plants in each crop have disease, but the severity overall is very low. So things are in control. Across region, East Central had the highest prevalence, so highest number of crops with disease, and Northwest and Southeast had the highest incidence. And the lowest prevalence, lowest incidence was in Northeast. Over years, we always had overall increase from 2012 to 2020 across the province. And we've been the only province that reported lower incidence of the disease in 2021, and then we got back to high. But Alberta just moved up, and Manitoba just moved up, but we reported lower, but we got back. So quite lots of crops and lots of plants with disease, but low severity. And then we go to verticillium stripe symptoms. So the new disease for the province, which is concerning for us, so we keep eyes on that. So verticillium stripe symptoms is usually chlorosis on one side of the stem. So if you are closer to harvest and you walk in your field and you see stripes up to one side of the plant, it's an indication of verticillium stripe. Early senescence, stunting of plants can happen. Chlorosis of lateral branches and leaves can happen. But this vertical yellow or brown stripe up one side of the stem is an indication of the verticillium stripe. There is one ultimate indication that no other disease shares with verticillium, because everything else is shared with at least Fusarium wilt, but Fusarium wilt is rare disease. But there is one symptom which is specific to verticillium stripe. And usually, unfortunately, it happens after harvest, so it's a bit too late. But the epidermis of the plant on the stem peels back, and it reveals lots of tiny structures, very tiny. So you need a magnifier to see them. And when you look at, with a good magnifier, when you look at these stems with epidermis peeled back, you will see abundance of very tiny black dots that they are microsclerotia. So they are very tiny. You need the magnifier to look at them, and that's only applicable to verticillium stripe. So if you see this is verticillium stripe. So a couple of comparisons, verticillium stripe versus sclerotinia, because both can have bleached stem, both can shred and shatter the stem, but the size of sclerotia is not even comparable with size of macrosclerotia. These are big. These are really big, so you can see them, and these are like the size of moss dropping and stuff. But these are very, very small. You need a good magnifier to see macrosclerotia on a shattered and shredded stems by verticillium. You will see better pictures. So comparing verticillium versus blackleg on the cross-section, Verticillium usually is just grayish and peppered throughout the cross stem, so it's mild, and there is no lesion with pycnidia associated with verticillium. But blackleg is solid, it's wedges, and usually there is a canker or a lesion with lots of pycnidia on it. So again, we don't look at only one symptom, look at everything. And if this stem peels back and I see macrosclerotia, then that's 100% verticillium. If this is a black wedge and I have a lesion with lots of pycnidia, that's 100% blackleg. And that's a clean guy. Fusarium wilt, sometimes people ask, because verticillium stripe is very similar in terms of symptoms to Fusarium wilt. The only thing different is that in verticillium we get the epidermis peeling back, and you can see macrosclerotia with a good magnifier. But on the other hand, we don't expect to see Fusarium wilt very often because we have the resistance, so it is rare to find Fusarium wilt. If it is very early in the growing season and you see only a stripe on one side of the stem, nothing else, then only a lab can confirm. Then you can send it even to our lab in Regina, crop protection lab, or any other lab that can do testing for verticillium and blackleg, and verticillium and Fusarium, and they will tell you if it's verticillium or Fusarium. But we do this in crop protection lab in Regina, in the provincial lab. So again, Fusarium wilt is the most similar to verticillium, but Fusarium wilt doesn't have that character of epidermis peel back and revealing tons of macrosclerotia. Again, more pictures from Fusarium wilt from Lyle. And again, this is the ultimate indication for verticillium wilt. So if it is shattered and shredded, epidermis peeled back, and you see abundant number of macrosclerotia, especially if you have a magnifier, then that's verticillium. So verticillium with typical signs of the disease is very new to the province. I still have time? Yeah. I guess I'm halfway through. 2 minutes? Okay, so I need to be very fast. So verticillium stripe is quite recent for Saskatchewan. We only have the visible signs of the pathogen, which are those macrosclerotia reported in Saskatchewan only from last year. And it was a farmer just combining the crop and just seeing something not expected, sending it to us, and it was just verticillium. And these are the macrosclerotia under the lens of our stereo microscope, and this is the exact sample that we received from that producer. So it was the first confirmed case of verticillium stripe with visible signs in Saskatchewan, and 2 lineages of the verticillium longisporum were found in those samples for very first time from Canada. So last year, in addition to general canola disease survey, we also had one verticillium-specific targeted survey. So a group of us went to the extreme east side of the province, close to Manitoba border, and purple was mine, and orange was Corey Jacob, our oilseed specialist who is here, and looked at 25 fields with 25 fields with, I can say, more likelihood of the disease being present. This is one of the patches of verticillium in very east side of the province. And this is unexpected data we got. So 16 of those 25 fields had verticillium. So quite a high number, but this is not, by no means, it is not representative for Saskatchewan, because this was a targeted survey. This was not a random survey. We targeted areas we believed, due to proximity to Manitoba, they may have disease, and unfortunately it came true. So 64% had the disease. So these were kind of more important diseases, Sclerotinia blackleg and verticillium. We touched quickly on clawroot later, but we also have other diseases like aster yellows. So if you see pods that they don't look pods anymore, they are oval and bladder-like, then that's an indication of aster yellows, which is a phytoplasma, which is a kind of a prokaryotic organism. And if you see your plants are purplish or red, and you will see structures where you don't expect, like you will see leafy structures, green leafy structures instead of yellow flowers. So that's phytoplasma, that's aster yellows. And again, pods are not like pods, are oval and like bladder. So just moving, 26% of our crops had in Saskatchewan aster yellows. We don't know what's the relationship with yield loss. No one has the yield loss model at the field scale so far. AFC works on an individual plant scale, but we don't know what's the impact at the commercial field. Alternaria is everywhere on every plant on the earth, perhaps. You can see it on the pods. 50% almost of the fields in Saskatchewan had Alternaria on pods, and these are usually very easy to diagnose because they are just oval, round. Still, I didn't get the yellow card. So I am supposed to get a yellow card and a red card later. But still, oh, yellow came. So we go faster. So brown girdling root rot, I have never seen them in the survey, but producers sent samples to our lab. This is rhizoctonia. This is diamond-shaped lesions on the crowns and lesions, or round lesions. And usually we isolate both rhizoctonia and fusarium out of it. And we don't know the border between brown girdling root rot and foot rot. Because if you get also foot rot, then you will get the same rhizoctonia and fusarium. So if it is fusarium, you may see the lesions may be pink. Again, these are not common in the province. We only got some samples from producers. We never saw them during the survey. And also these are not very well characterized diseases. So still researchers need to take a closer look at them. But if you saw a lesion that has none of the characteristics of sclerotinia or blackleg, looks a little bit pinkish, then think of root rot. Think of foot rot because it may be foot rot by fusarium. Gray stem, it's after harvest. It's a cosmetic disease, has no impact on yield loss, looks like blackleg. But if you scratch the lesion, the tissue beneath the lesion is so strong. So there is nothing else there. So you'll see gray lesions with lots of pycnidia or dots, but if you scratch them, nothing else is there. So it is more saprophytic and has no impact whatsoever on yield loss. So two farmers called me and said when they are combining, they saw a white clot everywhere around them. That's powdery mildew. Again, we are not worried about that. It comes too late in the growing season, has no non-impact on the yield. Downy mildew and white rust, we don't expect to see them. So if you see bump, pustules, white, just let us know. Breeders taking care of this. We should never be able to see these two. Red came. So clubroot, because you know clubroot, all of you, the clubs, I just give you the numbers we saw. So these are rotten clubroots, and these are some look like symptoms, but I jumped to the numbers. We looked at 521 fields, very well in control. In Saskatchewan, only 2 more commercial canola fields with visible symptoms, only 4 more detected through DNA. 3 new RMs added to the map. So it's 4 RMs, 1 with visible symptoms and 3 with pathogen DNA detected. And that's it. Thank you very much. So I couldn't go that fast.
Okay, thanks, Alireza. In the interest of time, we'll keep moving on to the next speaker, but if you do have any questions for Alireza, I do encourage you to connect with him at the break.